Medicine Without Cruelty presents organoids. So, what are organoids? Are scientific advancements which recreate biological relationships, but in a miniaturized, highly controlled laboratory setting. Now, you have learned about biological relationships in the previous learning outcome, where you learned about cells, tissues, and organs. So, that's what I mean about biological relationships. Because we understand that so well within human biology, we are able to create organ like structures. Now, organoids are 3D clusters of cells which are grown from stem cells. And stem cells, I haven't spoken about previously, but they are unspecialized cells which can develop into many different types of cells in the body. They're like the body's repair system, replacing injured or lost cells and tissues throughout life. Stem cells are very important because they act as templates. They have instructions for creating new cells. And it can be used in medical research because it can help explain the causes of serious conditions like cancer or birth defects. It may even be used as medical therapies to create cells and tissues for treating diseases such as Parkinson's, Alzheimer's, or even spinal cord injury. It can also be used to test drugs, so stem cells can be used to test new drugs for safety and effectiveness. Now, these organoids, coming back to organoids, they replicate the architecture and function of real human organs, including the interaction between cells and tissues. Now, this is important because in the lab, scientists use organoids to model specific human organs such as the brain, liver, or lungs. This allows researchers to study how diseases affect organs or how drugs interact with specific tissues, providing a accurate understanding of human biology. And there are many different ways that organoids can be used within science and research. Just like human tissues in the human body, organoids are made up of specialized cells which come together to mimic real organ structures. So, for example, a liver organoid contains liver cells that function similarly to how they would in the actual human liver, allowing scientists to, say, for example, test drug metabolism directly in this human like tissue. Just a case study here for you. Targeted therapy is a type of cancer treatment which focuses specifically on the unique features of cancer cells. Unlike traditional chemotherapy, which affects all rapidly dividing cells, targeted therapy is designed to identify and attack cancer cells with minimal damage to normal cells. Now, this is crucial because if we want to treat complex diseases like cancer, where people respond very differently to the same treatment, and remember, animals completely lack that genetic diversity that we find in humans. So, we need personalized treatment methods for treating complex diseases such as cancer. And a promising application of organoids in translational cancer research is the use of patients' fresh tumor tissue to establish patient derived cancer organoids for this personalized medication. Applications of patient derived tumor organoids range from preclinical modeling, drug screening, discovery, personalized medicine, among many, many others. Now, and researchers have developed patient derived organoids to create miniature replicas of an individual's tumor. Now, these patient derived organoids, or PDO, retain the unique genetic and histological characteristics of the original cancer, allowing scientists to test various drug responses in vitro meaning within a laboratory or a laboratory condition. This approach enables the identification of the most effective treatment strategies for each patient, thereby enhancing the precision of cancer therapy. Now, this case study by Crown Bioscience demonstrated the advantages of using PDOs for high throughput drug screening in cancer research. It highlighted that assay ready organoid platforms could reduce timelines, create high repeatability, and are suitable for extensive panel assessments, thus streamlining the drug discovery process. And it exemplifies the potential of organoids in advancing personalized medicine, offering a transformative approach to cancer treatment by tailoring therapies to the individual patient's tumor profile. Now, there are many different applications of organoids, and I'm just going to go through a few today. So, firstly, disease modeling. So, you've already seen precision or personalized medicine, but also By growing organoids from a patient's cancer cells, researchers can identify which drugs the tumor is resistant to. In addition to that, in oncology, we can provide combination therapy testing. So, organoids can help determine the most effective combination of therapies for individual patients. It can be used in infectious diseases. So, organoids can model by pathogens such as SARS-CoV-2, COVID-19, the Zika virus, and Helicobacter pylori, allowing scientists to observe how diseases interact with human tissues. It's applicable in neurodegenerative disorders, so that it can be used to study Alzheimer's, Parkinson's, autism by mimicking the neural networks of the human brain. And it can also be used in genetic disease research. So, for example, the organoid of an intestine was created from the intestinal cells of a person with cystic fibrosis and were then used to test various drugs to determine which drug would be most effective in that person. Organoids can also serve as a powerful platform for testing new drugs. For example, high throughput screening, organoids can be used to test hundreds of drug compounds simultaneously to identify potential treatments for diseases. It can be used to test drugs on an individual's organoid before even administering the drug, ensuring better outcomes and fewer side effects. And safety testing as well. Organoids can provide an accurate platform for evaluating the safety and toxicity of new drugs without using animals. Now, organoids hold real promise for repairing or replacing damaged tissues or organs. It can be used in liver regeneration. So, for example, if in liver failure, it could be used to implant healthy lab grown cells into the damaged liver tissue. Kidney organoids are being developed to study kidney disease and test potential regenerative treatments. And also retinal organoids, that's your retina in your eye. It can be used to understand and potentially treat blindness caused by retinal degeneration. And retinal degeneration, there are many different causes of that, both genetic and also environmental. And being able to repair your retina can potentially treat an individual's blindness. Toxicology testing. Now, organoids provide a human relevant platform to test the effects of chemicals, cosmetics, and pharmaceuticals. So, for example, for chemical safety testing, skin organoids are used to evaluate the effects of cosmetic products and chemicals, thereby removing the need for animal testing. Drug induced toxicity, liver and kidney organoids are particularly very valuable for predicting toxicity and side effects because the liver and the kidney, such as hepatotoxicity and nephrotoxicity. Now, drug induced liver injury is one of the leading causes of why drug tests may fail. And so, if we can provide valuable toxicity predictions, we know whether a drug is going to affect our liver. And organoids are also being utilized to assess the impact of environmental pollutants on human health. For example, whether they're airborne, and lung organoids can be used to study this on the effect of air pollution on respiratory health, and also water contamination. Kidney organoids can test the effects of toxins within drinking water, for example. It can also be used for studying human development and the microbiome. Now, organoids can provide a model to study the early stages of human development, such as organ formation, and it can understand how cells differentiate into various organ systems during embryonic development. It's very useful in developmental disorders because organoids can mimic congenital defects, helping us to understand the origins of these disorders. Now, our organoids can also be used to study how the microbiome, now your microbiome is the collection of all the microorganisms living within your body, and how that interacts with human tissues. So you can have gut microbiota interaction. So using an organoid that mimics your intestines to explore how gut bacteria influences diseases such as inflammatory bowel disease or colorectal cancer. And also with your immune response, by exposing organoids to pathogens or microbiota, scientists study immune responses in the gut, lung, and other tissues. Artificial intelligence can empower organoids. For example, predictive modeling, AI can analyze data from organoids to predict human tissue responses to drugs or chemicals, and also automated high content screening. AI can help process large data sets from organoid experiments, improving efficiency and accuracy. Thank you for listening.